Infection and disease. The epidemiology of infection and disease is the scientific and medical study of the causes, transmission, and prevention of disease within a population. Modern epidemiologists can trace its origins back to John Snow, the English surgeon who studied the cholera outbreak in the Soho district of London in 1854. His paper on the mode of communication of cholera is a model of epidemiological detective work. By marking on a map where each cholera death had occurred, Snow showed they all pointed towards the Broad Street pump, which supplied drinking water for the victims, and John Snow realized this was the source of the disease. In the end, the outbreak was quelled by simply removing the handle from the pump. Epidemiologists typically ask three questions. What's the source of the outbreak? How is it being transmitted? And how does one prevent further spread? The host-microbe relationship. Infection refers to the multiplication of a microbe in a host and the competition for supremacy taking place between them. In reality, there are thousands of microbial species and viruses on and within some human body systems. For example, the upper respiratory tract carries a diverse array of microorganisms, including Streptococcus, Neisseria, Haemophilus, and Staphylococcus, as well as the fungi Candida. The skin is populated with primarily gram-positive bacteria, including Staphylococcus, Streptococcus, Corynebacterium, Propionibacterium, and Micrococcus, as well as the fungus Candida. The female reproductive tract is densely populated with Lactobacillus, Staphylococcus, Corynebacterium, Streptococcus, and Enterococcus, as well as the fungi Candida. The oral cavity has a dense and diverse microbial population, including Streptococcus, Treponema, Neisseria, Haemophilus, Lactobacillus, Staphylococcus, and Propionibacterium. The urinary tracts of both males and females carry several bacterial species. The female urethra may contain Lactobacillus, Corynebacterium, Streptococcus, and bacteroids. The male urethra may contain Corynebacterium and Streptococcus. The small intestines contain bacteroids, Lactobacillus, and Streptococcus, whereas the large intestines carry a dense and diverse microbial population. The 100 trillion microbes found on and in the human body that do not cause disease represent the microbiota, biota meaning life. The microbiota is a population of microorganisms and viruses residing in the body without causing disease. There are both the indigenous microbiota and transient microbiota. The indigenous microbiota establish a permanent relationship with various parts of the body. The transient microbiota are more temporary and are only found for limited periods of time. The relationship between the body and its microbiota is an example of a symbiosis, meaning living together. If the symbiosis is beneficial to both the host and the microorganism, this relationship is referred to as mutualism. If the symbiosis is beneficial to only the microbe and the host is unaffected, this is called commensalism. And if the symbiosis is beneficial to the microbe but the host is harmed, this is called parasitism. The colonization of microbiota in the human body begins either at birth in the case of vaginal birth or shortly after birth in the case of C-sections or cesarean births. During vaginal birth, the infant's gut becomes colonized with maternal microorganisms that can contain Streptococcus, Bifidobacteria, and Lactobacilli. Other species will then colonize due to contact with instruments and other people, and by feeding, whether nursing or formula. This leads to further colonization with high maternal oral and gastric bacterial species, leading to a gut colonized by 400 to 800 bacterial species. In the case of cesarean birth, this colonization is delayed, but the colonization begins with microorganisms the newborn encounters in the environment. These may contain a high amount of Clostridial species and low Bifidobacterial species. Contact with instruments and other people, and feeding with either nursing or formula leads to other types of colonization later on, usually including high environmental species such as Bacteroidetes and Enterobacteriaceae, leading to a gut colonized by 400 to 800 bacterial species. Pathogenicity refers to the ability of a microorganism to gain entry to the host's internal tissues and to bring about physiological or anatomical change, resulting in altered health and leading to disease. Whether a disease is mild or severe depends on the pathogen's ability to do harm to the body. Thus, the degree of pathogenicity is called virulence, viral meaning poisonous. For example, an organism invariably causing disease, such as typhoid bacillus, is said to be highly virulent. Pathogenicity islands is a term that has been used to refer to clusters of genes responsible for virulence called virulence factors. Virulence factors are pathogen-produced molecules or structures that allow the cell to invade the host or to evade the immune system of the host and possibly cause disease. An infection and a resulting disease can be caused by a single microbe, or it can be caused by two or more microbes acting together or in succession. Such polymicrobial diseases include tooth decay, gastroenteritis, urinary tract infections, otitis media, and HIV and AIDS. For disease to occur, a potential pathogen must first come into contact with exposed parts of the body. Several outcomes are possible. The pathogen may be lost to the environment, or it may colonize the normal microbiota and remain as a transient member. Depending on the nature of the pathogen, it could also become commensal. Commensal bacteria act on the host's immune system to induce protective responses that prevent colonization and invasion by pathogens. On the other hand, these bacteria can directly inhibit the growth of respiratory pathogens by producing antimicrobial products and competing for nutrients and adhesion sites. Infections can either be exogenous or endogenous. An exogenous infection is established if a pathogen from the environment breaches the host's external defense system and enters the internal portion of the host. An endogenous infection ensues when or if a microbial member of the normal microbiota gains access to internal sterile tissues. Opportunistic infections often are caused by commensals taking advantage of a shift in the body's delicate balance to one favoring the microorganism. If the indigenous microbiota is reduced or if the host's immune system is weakened, some commensals seize this opportunity and invade the tissues, causing disease. AIDS is an example where crippling of the immune system makes the patient highly susceptible to such opportunistic organisms. Thus, an upset in resistance mechanisms or microbiota control may enhance the ability of microorganisms to establish disease. Infections may develop in one of two ways. A primary infection occurs in an otherwise healthy body, whereas a secondary infection develops in an individual that is weakened by a primary infection. Diseases may be referred to as either local or systemic diseases. Local diseases are restricted to a single area of the body. Systemic diseases are those disseminating to the deeper organs and systems. For example, a Staphylococcus skin boil beginning as a localized skin lesion may become more serious when staphylococci spread and cause systemic disease of the bones, meninges, or heart tissue. Infection of the blood can be very serious, leading to death. Septicemia refers to an infection of bacterial cells in the blood, which can be life-threatening. If the pathogenic organism in the blood is a fungi, it is referred to as fungemia. Viremia refers to the spread of viruses through the blood, and parasitemia refers to the spread of protozoa and multicellular worms through the blood, all of which can be life-threatening. Establishment of infection and disease. Disease progression involves incubation, prodromal, acute, decline, and convalescent stages. A susceptible person could be exposed to the flu virus in respiratory droplets, at which time the incubation period begins. The prodromal phase is characterized by mild signs and symptoms, such as headache and fever. The acute period is characterized by sudden symptoms of high fever, chills, cough, tired muscles, joint pain, and loss of appetite. During the period of decline, the virus is eliminated from the body. The fever breaks and appetite returns as recovery begins. With the period of convalescence, the body returns to normal. Signs of a disease are defined as evidence of disease detected by an objective observer, such as a physician or medical staff member. Symptoms of a disease are defined as changes in the body that are sensed by the patient. Diseases also may be characterized by a specific collection of signs and symptoms called a syndrome. The incubation period is the amount of time that exists between the entry of the microorganism into the host and the appearance or onset of the first symptoms. The prodromal phase is the time of mild signs or symptoms. A clinical disease is one in which symptoms are apparent. A subclinical disease is accompanied by only a few obvious symptoms. The acute period or climax is the stage of the disease in which signs and symptoms are of greatest intensity. Examples would be the skin rash and scarlet fever, jaundice and hepatitis, or swollen lymph nodes in the infection of mononucleosis. The length of this period can be quite variable depending on the body's response to the pathogen and the virulence of the pathogen. During a period of decline, the signs and symptoms begin to subside. Sweating may be common as the body releases excessive amounts of heat, and the normal skin color soon returns as the blood vessels dilate. The period of convalescence is the time in which the body's systems return to normal. Diseases can also be categorized as either acute or chronic. An acute disease, like the flu, develops rapidly and is usually accompanied by severe symptoms, comes to a climax, and then fades rather quickly. A chronic disease, by contrast, often lingers for long periods of time. The symptoms are slower to develop, and an acute period is rarely reached. Convalescence may continue for several months. A portal of entry refers to the characteristic route by which an exogenous pathogen enters the host. In order for a pathogen to cause disease, it must gain entry into the body. A portal of entry refers to the characteristic route by which an exogenous pathogen enters the host. The portal of entry varies considerably for different organisms and is a key factor leading to the establishment of the disease. Abrasions or mechanical injury to the skin can be a portal of entry. The amount of pathogen needed to cause a disease varies. This is referred to as the infectious dose. The infectious dose reflects the ability of a pathogen to establish an infection and cause disease. The ability of a pathogen to penetrate tissues and cause structural damage is referred to as invasiveness. Invasiveness often is facilitated when the pathogen becomes internalized through phagocytosis by immune cells. After entering a host cell by phagocytosis, the bacterial cell loses the surrounding membrane and divides to form a larger population. Invasion of neighboring cells uses the thin actin tail to drive the cells into the adjacent cell. That cell again loses the surrounding membrane and initiates another infection, repeating the cycle. The virulence of a microbe depends to some degree on its ability to produce a series of extracellular enzymes designed to help the pathogen resist the body's immune defenses. One of these enzymes is coagulase. Coagulase catalyzes the formation of a blood clot from fibrinogen proteins in human blood. The clot sticks to staphylococci, protecting them from phagocytosis. Part of the walling off process observed in staphylococci skin boils is due to such a clot formation. Streptokinase is an enzyme that dissolves fibrin clots used as a defense by the body to restrict and isolate the infected area. Streptokinase thus overcomes an important host defense and allows further tissue invasion by bacterial cells. Hyaluronidase is sometimes called the spreading factor because this enzyme enhances penetration of the pathogen through the tissues. This enzyme digests hyaluronic acid, which is a polysaccharide that binds cell tissues together. Microbial poisons, called toxins, can profoundly affect the establishment and course of a disease. The ability of pathogens to produce such toxins is referred to as toxogenicity. Toxins present in the blood is called toxemia, and the person infected is considered intoxicated. Two types of toxins are recognized: exotoxins and endotoxins. Exotoxins are heat-sensitive protein molecules manufactured during bacterial metabolism. Endotoxins are present in the outer membrane and are part of the lipid polysaccharide complex. Pathogens must be able to also leave the host. There are 6 different portals of exit from the body. This includes coughing and sneezing, through an open lesion, in the urine or feces, by an insect bite, or through removal of the blood. Infectious disease epidemiology Infectious disease epidemiology is concerned with how infectious diseases are distributed in a population and the factors influencing or determining that distribution. To cause an infection, pathogens have to be transferred from a source to a susceptible host. Places where a disease-causing microorganism is commonly found is referred to as a reservoir. Animals are one type of reservoir. Water and soil are other types of reservoirs. A special type of reservoir is a carrier. A carrier is a person who has recovered from the disease but continues to shed disease agents, possibly infecting other people. Diseases can be described as communicable or non-communicable. Communicable diseases are infectious diseases transmissible or contagious. They are relatively easily passed on from one host to the next. Non-communicable diseases are singular events in which the infectious agent is acquired directly from the environment. Non-communicable diseases are not easily transmitted from person to person. For example, in tetanus, penetration of soil contaminated by Clostridium tetani spores can enter into the anaerobic tissue of a wound from where the disease develops. Tetanus is not spread person to person. Infectious diseases can be transmitted in several ways. One of these is direct contact, also known as person-to-person or horizontal transmission. Here, a close or personal contact with somebody who is infected or who has the disease spreads the illness. Handshaking or kissing an infected person can spread bacterial cells, viruses, and other pathogens to an uninfected person. Direct transmission also can involve the violent expulsion of respiratory droplets through sneezing, coughing, or simply talking. Droplet transmission through the air requires the recipient to be relatively close to the infected individual. In a sneeze, the droplets can travel 150 feet per second. However, the droplets are fairly large and fall out of the air within about 1 meter of their source. If an uninfected person is within that distance, the eyes, mouth, or nose may be portals of entry for such an airborne pathogen. Direct contact, called vertical transmission, includes the spread of pathogens such as HIV or Toxoplasma gondii from a pregnant mother to her unborn child. Indirect contact can be the result of contact with a non-living object or medium or a vector such as an organism. Fomites are inanimate objects on which or in which disease organisms may linger for some period of time. Examples are doorknobs, countertops, restroom stalls, and sinks. Vehicle transmission involves the indirect spread of disease through contaminated food, water, or air. Vectors can be of two types, either mechanical vectors or biological vectors. Mechanical vectors represent arthropods that passively transport these microorganisms on their legs or other body parts. Biological vectors is where the pathogen must multiply in the insect or other organism before it is able to infect another host. Diseases also are described by how they occur within a population. Endemic refers to a disease habitually present at a low level in certain geographical areas. An epidemic refers to a disease that occurs in a community or region in excess of what is normally found within that population. An outbreak is a more contained epidemic. A pandemic is a worldwide epidemic affecting populations around the globe. Nosocomial infections represent that portion of healthcare-associated infections that are associated with hospitals. Nosocomial infections account for an estimated 1.7 million infections, and according to the CDC, almost 100,000 deaths each year in the United States. Like all infections, nosocomial infections involve three elements. 1. A compromised host, such as a hospital patient. 2. A source of hospital pathogens. And 3. A chain of transmission. Medical facilities can be a reservoir for human pathogens. The majority of nosocomial infections are caused by opportunistic agents. Opportunistic agents are microorganisms that do not normally cause disease in individuals, but in ill or immunocompromised patients, given the opportunity, an infection can occur leading to disease. Potential pathogens can spread through several means disease within the hospital or healthcare environment. Protecting patients and other healthcare workers also means using standard precautions if working with a patient who might be infected. The precautions when handling blood or other bodily fluids that may harbor pathogens include washing hands, wearing personal protective equipment, handling and disposing of sharps properly, disposing of all hazardous and contaminated materials in approved labeled biohazardous containers, and cleaning up all spills with disinfectant or diluted bleach solution to kill any pathogens that may be present. Numerous reasons help explain how disease emergence and resurgence are driven. This can include changes in land use or agricultural practices. Urbanization, deforestation, and water projects can bring new or re-emergent diseases such as dengue fever and schistosomiasis. Also, changes in human demographics can be a factor. The migration of many peoples or whole societies from agrarian to urban lifestyles has brought about new diseases to a susceptible population, such as is seen in malaria. In many developing nations, large numbers of people suffer from malnutrition or poor public health infrastructure, making disease eruption much more likely. There is also the matter of pathogenic evolution. Pathogens have developed resistance to antibiotics and antimicrobial drugs, leading to the resurgence of many diseases, including tuberculosis and yellow fever. Contamination of food sources and water supplies is definitely a factor. Substandard application of or lapses in sanitation practices can bring resurgence of disease, such as is seen in cryptosporidiosis. In COVID-19, we have seen that international travel assists in quickly spreading disease. The number of people traveling internationally can spread diseases to other parts of the globe very quickly, as was observed with SARS, COVID-19, West Nile virus, and the swine flu. Also, failure of immunization programs can bring about the resurgence of certain diseases, such as diphtheria. International trade is also a factor. The global movement of produce can introduce new or resurgent diseases, such as hepatitis A and cyclosporiasis. Likewise, wildlife trade, whether legal or illegal, provides new mechanisms for disease transmission, such as is seen in monkeypox. Climate change is another factor that should be looked at. Global changes in weather patterns bring new diseases to new latitudes and elevations, leading to resurgence of diseases such as hantavirus pulmonary syndrome. The CDC has had a history of serving American public health. Regarding emerging and resurgent diseases, the CDC's priority areas include the following: international outbreak assistance to host countries to maintain control of new pathogens when an outbreak is over, a global approach to disease surveillance by establishing a global network of networks for early warning of emerging health threats, applied research on diseases of global importance, global disease control through initiatives to reduce HIV disease, AIDS, malaria, and tuberculosis, and public health training that supports the establishment of international emerging infections programs in developing nations. Thank you for watching.